Our speaker today is Bernard Lerer from Hadassah, Hadassah Hospital and the Hebrew University. He works on the molecular genetics of psychiatric and movement disorders and the neurochemical mechanism and clinical psychopharmacology of antidepressants and brain stimulation. And today he will tell us about schizophrenia in Arab Israelis. Please.
Well, thank you. First of all, I must protest vigorously that I'm forced to talk in English. It's very difficult for me. I'll do my best to communicate with you in my broken South African dialect. Secondly, it's a real pleasure to be here. I know quite a few people in the room, but from different circumstances, different places, and different committees of the university. So it's nice to be here and meet you on your home turf. I got an email from Chagai Bergman, whom I saw last in the middle of the night during the Har La'evek race, that he would have come, but unfortunately he's overseas. And he's really the only one that I actually do scientific collaboration with out of this august group. The second point, I think it's important to make what is called in Hebrew a gilui naot, and that is that I think there's one area called neural computation, which you know a lot more than me about. I think I know much more than you about psychiatry, certainly about clinical psychiatry, and probably most of us share the fact that we don't know all that much about genetics. So I think we're on sort of an even turf here. I'm going to talk a little bit about the studies we've been doing for several years, looking for susceptibility genes for schizophrenia. We've looked in both the Jewish population of Israel and the Arab population, but I think that the studies we've done in the Arab population really are particularly interesting because of the homogeneous inbred nature of the sample that we've studied, and I'll tell you a little bit more about that as we go along. So what I'm going to do is I'm going to start with a general background on schizophrenia, in case there are some of you who are not familiar with this particular illness. I'll then talk a little about something which is more history than current science, but I think quite a glorious chapter in the history of psychiatry, and that is in the pre-molecular era, the attempts to successfully establish that schizophrenia is indeed a disorder with a major biological and a major genetic component. Even at the time when I began my residency in psychiatry, there was still a strong belief in the psychoanalytic causation of psychiatric disorders, even serious brain disorders like schizophrenia. And then, of course, the bulk of the talk will focus on our attempts to locate genes that are responsible for schizophrenia. So first of all, what is schizophrenia? It's a serious psychiatric disorder which is characterized by three types of symptoms: what are termed positive, negative, and cognitive symptoms. The positive symptoms are the dramatic symptoms of schizophrenia. They're not positive in any way, but they're termed positive because the philosophy is taken from the definition of Ewlings-Jackson in relation to epilepsy. They refer to the hallucinations, the delusions, and the formal thought disorder, the manifestations that make schizophrenic patients appear crazy or mad. The negative symptoms are, of course, the most serious. The positive symptoms generally respond to medication. The negative symptoms are symptoms of avolition, of withdrawal, of difficulty in performing daily tasks, difficulty in experiencing pleasure, anhedonia, what is termed abulia, a kind of a lack of will, a lack of ability to get down and do anything. And these are the features of the chronic form of the disorder. And this is what is the most devastating part of the disorder, because this is why schizophrenia is a chronic illness, as opposed to, say, bipolar disorder or manic depressive illness, where patients can achieve a substantial remission with appropriate treatment, but then are able to function normally. Schizophrenia patients, for the most part, are never able to function normally. They're also not able to function normally because they have significant cognitive impairments, primarily impairments of executive function, difficulty in performing more than one task at the same time, attention, memory, and also a slight, not severe, deterioration of intellectual functioning. Kreplin, who was the one who first described schizophrenia, called the illness dementia precox, which is dementia that comes on at an early age, but there isn't really no dementia per se in schizophrenia. How common is schizophrenia? Very common. It has a 1% lifetime prevalence. The age of onset is in the second and third decades, so we're talking about late adolescence, early adulthood. So the public health impact of this illness is enormous. At least 1/3 of schizophrenia patients are chronic, and by current definitions, which require the illness to manifest for at least 6 months before the diagnosis is given, it's actually more like 80% of schizophrenia patients develop a chronic illness. To the lay public, one of the most famous individuals with schizophrenia is John Nash. The difference, incidentally, between bipolar disorder and schizophrenia manifests in the number of famous people who have the illness. If you go to the internet and you look for famous people with bipolar disorder, you'll get a list of hundreds and hundreds of people, from Abraham Lincoln to Ted Turner, the founder of CNN. But if you look for schizophrenia patients who are famous, you won't find very many. John Nash is one of them, and many of you probably saw the movie A Beautiful Mind, which describes, I think, vividly the positive symptoms of the illness and then the slide into the negative features of the disorder. Nash became ill when he was a young scientist, and he was kind of rediscovered when it was decided to award him the Nobel Prize. Besides the movie, which I think is very interesting, I saw on Israeli TV actually a interview with Nash, which I think is absolutely fascinating. So I took a piece of it for you.
You can't
That's Nash and his wife.
I've learned that it's better that I don't talk about it.
As for questions about Nash as a father, after years of estrangement, he says he's now close with an older son he has from a previous relationship. He even arranged for a cameo appearance for him in the movie. He's the orderly on the right. He also shared with him 1/4 of the money he got for selling his story to the filmmakers. Another quarter went to his and Alicia's son Johnny, who shares more than just a name with his father. He also shares a talent for math. Johnny earned his own PhD in 1985, and, like his father, he suffers from schizophrenia.
What kind of a father was he?
Well, he wasn't around for the first 10 years of my life, but when he came back at age 10, he was a very good father, yes.
Was there any doubt in your mind that you wanted to be a mathematician?
Oh, no, there was no doubt at all, yes.
You wanted to follow in your father's footsteps?
Absolutely. To do honor to Dad, follow in his footsteps, that was always my life's dream. I still felt that I could equal him, that I could, I haven't done so yet, but I'm trying to.
He could do a lot if he would really think clearly. What he really needs is to get his thinking in order, more rational.
More rational.
Rational.
Rational thought.
Yes. Like a diet.
Like a diet. And goals and appetites.
So what's interesting, if you noticed in the movie, is that the son, unlike his father, has a movement disorder, tardive dyskinesia, which is a consequence of the use of long-term use of antipsychotic drugs, and not a feature of the schizophrenia per se. His father also was treated with antipsychotic drugs, and why he didn't get TD whereas his son did is, of course, another question for another talk. What do we know about the neuropathology of schizophrenia? The answer in the next slide is really the answer. We know very little. Schizophrenia has long been called the graveyard of neuropathologists because so many of them have studied it for so long and actually found nothing. very little microscopic pathology of the brain has emerged. What is clear that there are brain changes, and this is a work from one very interesting study published now almost 10 years ago, which looked at young, early onset schizophrenics. Early onset schizophrenia at the ages of about pre-adolescence or very early adolescence is an even more severe form of the illness than adult onset schizophrenia. Schizophrenia which starts in late adolescence and early adulthood. And you can see in comparing the normal adolescents to the schizophrenia subjects, you see a very clear loss of volume which affects parietal, frontal areas of the brain. It's a kind of a diffuse loss of brain volume. It's progressive. It is related somewhat to the use of antipsychotic drugs, but not directly and not fully related. But overall, really, there is no clear diagnostic neuropathology, which of course makes it even harder to understand the biological causation of the illness. Just before we go into the biology, just one more word about the clinical aspects. How do we diagnose schizophrenia? We want there to be present at some stage of the illness delusions, hallucinations, disorganized speech, grossly disorganized or catatonic behavior, or a kind of apathy or lack of emotion. We need to see social occupational dysfunction, needs to be present for at least 6 months, and not to be due to substance use or abuse. The classical treatment of schizophrenia is with neuroleptics or antipsychotic drugs. All known antipsychotic drugs bind to and block dopamine D2 receptors, and one needs an occupancy of about 60% of dopamine D2 receptors in order to have an antipsychotic effect. There is no antipsychotic drug known to date which does not block dopamine D2 receptors. There's been excitement over the years and papers in Nature Neuroscience, Science, etc., which claim to have developed antipsychotics which do not block D2 receptors, but these have never progressed to a stage of replication in larger scale clinical trials. So there is something key, something critical about the dopamine system, and particularly the D2 receptor. There are two types of antipsychotics, the typical and the atypical. The atypicals differ because they block additional receptors and they don't have such severe extrapyramidal symptoms, but they do have a whole host of problems of their own, like diabetes, weight gain, hyperlipidemia, so they're certainly not the answer. In terms of theories of schizophrenia, there are several, as is inevitable for an illness for which no cause is really known. The neurochemical hypotheses essentially derive from the knowledge we have of the pharmacology of the illness. They essentially posit that dopamine D2 receptors or dopamine overactivity in key areas of the brain underlies the illness. This being, of course, derived from the fact that D2 blockade improves psychotic symptoms, which is almost like saying that a headache is an illness caused by lack of paracetamol, and that is the underlying causation of headache. But there is a fair amount of evidence from studies post-mortem, from imaging studies using D2 receptor ligands, and D1 receptor ligands, and there is a fair amount of evidence, but it's not entirely consistent, and the difficulty is disentangling the evidence from the effect of antipsychotics, because in vivo studies or post-mortem studies are inevitably done in patients who've had quite a long exposure to drugs which affect the dopamine system. So it's very hard to disentangle the effect of the drugs from the effect of from the illness itself. And the answer, of course, is to study first episode patients who've never had antipsychotic treatment, but it's not easy to recruit such patients into studies. It's not easy to find them, and if you do find them, it's very hard to have them in a PET scanner or in an MRI for very long. So this is an exceedingly difficult challenge. A second line of theories is the neurodevelopmental approach, which is, I think, the more current of the theories, which posits essentially that schizophrenia is not an illness that begins at the age of 18 or 16. That essentially the core or the basis of the illness is laid during in utero, during brain development in utero, and that the illness manifests acutely at the age of late adolescence or early adulthood, but there are prodromal signs present a long time before. We don't have the time now to look at this kind of evidence, but there are intriguing studies looking at performance of children on earlier examination, children who later became schizophrenic on examinations done at the age of 11 or 12, the 11 plus exam that used to be in England, the MILTA, which is done in Israel, and also some excellent studies from Israel by a group which has been looking at the testing done in the recruitment center, in the Schizophrenia, and looking at individuals who subsequently became schizophrenic for early signs. So there's a lot of evidence to suggest that there are abnormalities of function, that there may be slightly lower overall intellectual function in schizophrenia patients. There may be a greater tendency to abuse substances. A lot of studies from Scandinavia suggest that there's a tendency to greater cannabis use, which may be a consequence of the developing illness or a cause of it. That's not clear. And there's another line of studies which posits a immunological basis for the disorder. A lot of evidence, a lot of work done, but nothing definitive that emerges.
Are these mutually exclusive?
No. They're certainly not mutually exclusive, and clearly they could be they could cross each other in different ways. Just for convenience, I summarize it that way. We and many other people have been interested in the genetic basis of the illness, and before talking about that, I always put up the slide, which is from one of the Jewish families that we've been studying. A female with schizophrenia, married to a not very vocationally successful man without any formal illness, though. And if one looks at the children, one sees everything here. One sees significant number of schizophrenia patients. One sees children who were brought up at home and didn't develop schizophrenia. One sees children who were brought up in an environment entirely outside the home and did develop schizophrenia. So clearly we're talking about a complex inheritance. We're not talking about a Mendelian disorder, and I'll show you the actual evidence for that in a few moments. But just looking at the genetic risk for schizophrenia. What is the risk for schizophrenia? The lifetime risk. In the general population, it's 1%. So 1 in 100 individuals will develop a schizophrenic illness during the course of their lifetimes. Not all will become chronically ill. If one looks at identical monozygotic twins, the risk is less than 50%. And there have been many studies looking at this, and the risk is about 48%. So significantly greater than in the general population, but still half of the individuals who are monozygotic twins who develop schizophrenia, in half the cases the co-twin does not have schizophrenia. Yes?
Any among those who were reared separately for one reason or another?
There are studies, but they're not enough. They're not enough studies to really make it definitive. The classical twin studies in schizophrenia have been comparisons of monozygotic and dizygotic twins. And if you can see fraternal twins, the rate is 17%. So essentially the difference between the rate in identical twins and in fraternal twins represents the genetic component of the illness, since the environment for the most part of fraternal and identical twins is shared if they're brought up together. And one can roughly assume, it's not entirely true because there is non-shared environment as well in identical twins, but roughly speaking, this is the heritability component of the illness. About 80% heritability is calculated for schizophrenia. Next highest, are two, this isn't for some reason, it's not on this version of the slide, but with two parents with schizophrenia, the risk is about 45%. If two individuals have schizophrenia and they have children, the risk amongst the children is about 40%. And for the rest of the relatives, it's kind of spread in between. But you can see that it drops off precipitously as one goes down in terms of the strength of the relationship. So for first cousins, it's only about a 2% lifetime risk as opposed to a 1% in individuals with in the general population.
It's not symmetric between uncles and nephews?
No.
So children and parents.
Or children and parents?
Yeah. It is not, and I don't give really an explanation of why that is the case. This is data from about 100 studies, I would say, that is put together by Gottesman in 2001. So I mentioned the difference between monozygotic and dizygotic twins as being reflecting the genetic liability, and of course the discordance within monozygotic twins reflects the environmental effect. Just one really nice study done by Gottesman in 1989 looked at the offspring of the monozygotic non-schizophrenic co-twin. So if you have a twin with schizophrenia and a twin without schizophrenia, they're monozygotic, what happens with their children? And the finding is clear that the rate of schizophrenia in the children of the unaffected co-twin is significantly increased. So that whatever genetic factor is present and is not penetrant in the non-ill twin manifests itself to a greater extent, for whatever reason, in the offspring of that twin. This is a fascinating study, but as you can see, the numbers are very small. It's not easy to identify these kind of people. And just as a tribute to Seymour Ketty, who was probably one of the pioneers of psychiatric genetics, the Danish adoption studies, which were really extremely important in showing the frequency of schizophrenia in the biological parents of children adopted away at birth. And what was really found is that in the schizophrenic adoptees, the rate of schizophrenia is far greater than amongst the control adoptees' parents, even though these were children who were adopted at birth and never ever lived with their parents. And finally, environmental factors. What environmental factors do we know? If 50% of the risk in monozygotic twins is environmental, what environmental factors do we know? The answer is not all that much. There's evidence that maternal stress and hunger, influenza events during pregnancy. Recent studies suggest that advanced paternal age is a risk factor for schizophrenia. Certain complications of pregnancy, early rearing environment, urban birth, and adolescent cannabis use. These are a few of the factors which have been linked environmentally to risk for schizophrenia. But there are no studies, no comprehensive studies of gene-environment interaction in schizophrenia. Some of you may have seen the studies of interaction between certain genetic factors and childhood maltreatment done by Caspi in New Zealand, but these did not relate to schizophrenia. These related to later risk of depression, not later risk of schizophrenia. So there are no gene-environment interaction studies published to date which are substantial in any way. So we're talking then in schizophrenia about a complex inheritance model. We're talking about several genes which are most likely involved. It could be that there are many common genetic variants which are playing a role. It could be that there are rare mutations, and there could be epigenetic mechanisms at work as well. What is clear is that there is not a single major locus. There is no single major locus that underlies schizophrenia. Very, very, very few pedigrees, if any, have been described which convincingly show a schizophrenia-like illness attributable to a single major locus. So what remains to be established is, are we talking about many different common variants which interact, or are we talking about a large number of possibly even an impossibly large number of very rare mutations. And this is the current thinking, and I'll come to it in a few minutes. Whether that thinking is by elimination because of the inability to find the genes thus far or not is something that one has to ponder. How have people sought to find the genes? Initially, in the beginning of the molecular era, they did linkage studies, which were based on co-inheritance of disease markers and the disease in families. Linkage studies are much more attuned, much more suited to Mendelian disorders, but there was a lot of hope in the 1980s that they would get us somewhere. Association studies comparing the prevalence of certain alleles in patients versus those from the same ethnic group. Expression studies comparing mRNA expression pattern in patients versus controls. CNVs, which are essentially larger variations in copy numbers, variations in chromosomal variations, which can range from 1 kilobyte to several kilobase to several megabases. And now the focus is in sequence analysis. All these are indirect methods. The focus now is on sequence analysis, and these studies are going to be emerging in the next few years. I'm not going to talk about the results of sequencing because we don't have them as yet, but we, like others, are engaged in directly sequencing patients with schizophrenia. Overall, there have been two major approaches. A hypothesis-driven approach, whereby candidate genes have been studied. So obviously, a strong candidate would be the dopamine D2 receptor. It would make sense that if antipsychotics block the D2 receptor, if we have a genetic variant in the D2 receptor, that is an ideal place to look. And of course, many other receptors like serotonin receptors, glutamate receptors, and so on, have been the locus of a search for variants that underlie schizophrenia. The more widely used approach has been a hypothesis-free approach, searching for schizophrenia genes without any a priori hypothesis, using genome-wide methods, using genome-wide association studies, using sequencing, whole genome sequencing or exome sequencing. The concept being that we have really no idea what the pathophysiology of the disorder is, and we will look for genetic variants associated with the disorder, and once we find them, we will then attempt to understand what their significance is and how they might be related to the illness pathophysiologically. And that's the approach that we used, and which I'm going to talk to you about in a few minutes. So there have been a lot of genome-wide association studies of schizophrenia using single nucleotide polymorphisms. Recently, there's been a spurt of results focusing on the HLA region, which look very promising. But I think that on the MHC region, there have been 3 findings in this region from different studies. But it's really, I think, clear that no strong candidate has emerged. No clear association of any gene with schizophrenia has emerged from the GWASs that have been done to date. Nor thus far with copy number variations. So in summary, thus far, there is no diagnostic neuropathology of schizophrenia. The mode of inheritance is most likely multifactorial. An unknown number of genes are involved. There's an interaction with environmental factors. There are a large number of variants, including common variants as well as rare variants. And, well, the final point is obvious. So what have we contributed to this ongoing saga? We studied in probably one of the more fortunate decisions I've made in my scientific career. We decided to focus on a group of villages in the Mishulash, served by the Taibe Regional Mental Health Center. Why did we focus there? Well, on the one hand, for simply administrative logistic reasons, because the head of the Taibe Mental Health Center is still an Arab Israeli psychiatrist with enormous interest in genetics, so he was happy to collaborate with us. and also because we wanted to focus on a region where we could get to as many of the families as possible. We wanted an ethnically homogeneous group, and Taida has an ethnically homogeneous population, but not only is it ethnically homogeneous in the sense that the Arab-Israeli population is, but it's a population which was founded less than 100 years ago, which had rapid population growth, a very high level of consanguinity. About 25% of the marriages in the region of the Mashulash are within families or first cousin marriages, and a very low rate of intermarriage with other population groups, right up to this day. And very large families. These are examples of some of the families that have been recruited from this area. So you can see large families with many affected and several generations. So these are really an ideal population to study and to seek genes for major psychiatric disorders such as schizophrenia. So our first venture was to do a whole genome linkage study. The genotyping was actually done right here in this building by Mira Korner. It was the first linkage study, the first whole genome linkage study with microsatellite markers actually done in Israel, and the results were really very intriguing. So we were then looking not for specific genes, but for chromosomal regions which might harbor genes that are associated with the illness. And the strongest findings were on chromosome 6q23, where we had a very highly significant association, LOD scores in the region of in excess of 4, where one requires a LOD score of 3 to declare a positive linkage. So this was the strongest associated region. The second strongest was the 2q region, where the linkage was not significant but suggestive and quite strongly so, and then also the 7p region and the 10q region. So we emerged from this whole genome linkage study on 18 families, which had 88 individuals with DNA, of whom 48 were affected. We emerged with 3 strong areas of linkage, the strongest being 6q23. So this was about 12 years ago, and it was at that time the possibilities available for following up were limited. One could do SNP association studies, single nuclear polymorphism association studies. That was the only way of doing fine mapping. One couldn't do large-scale studies, and so what we did was we decided to focus on the 6q23 region. And this work was done by one of my PhD students, Daniela Aman, a student who was my student and Doron Lancet from the Weizmann Institute. We supervised her together. And she focused on mapping this region, the 6q23 region. And what emerged was a very strong association. There were some sort of isolated peaks of association. This shows the minus log p-value, and this is the cutoff for multiple testing. So any finding that is above this line is a finding which is significant beyond correction for multiple testing. And what we found was in the region of a gene called AHI1, the Abelson Helper Integration Site 1 gene. There on chromosome 6q23 we had our strongest finding, and adjoining this a gene called an open reading frame called 660 or F217, a gene not yet described. So here we had the strongest association, and these were suggestive, but adding SNPs in these regions did not give us additional significance. And when looking at the haplotypes, which is essentially looking at several SNPs together, you can see that the finding is even stronger and more focused on AHI1. And so this was a very exciting finding, which we published in 2006. And to our enormous delight, there were several replications published soon after, continuing right up to last year. An unusual situation in psychiatric molecular genetics. The first publication came out very soon in 2007. This was in an Icelandic population, which a work done by the Decode company in Iceland. And then a very large European consortium published a replication showing support for the same markers, the same SNPs in the AHI1 gene. And finally, a paper that came out, I think, last year, which showed association as well. This was in samples from Germany and Spain. So we see that this, even though we discovered this association in a highly inbred Arab-Israeli population, it was demonstrable thereafter in Icelandic patients, in patients from Spain, patients from Germany, and also patients in samples taken from the whole of Europe. Now, a lot of you will probably ask, maybe not right away, but when you go home and think about it, if that is the case, why have the genome-wide association studies not shown association with AHI1? One of the most famous associations with diabetes is the TFCL6 polymorphism, which emerged from this type of linkage study followed by association. And then when they started doing GWASs, every single GWAS that appeared, this particular SNP was the strongest association in that GWAS. This is not the case with AHI1, which means that the most likely explanation is that this gene contributes to schizophrenia, but it does not contribute to all cases of schizophrenia. It may contribute to a subtype of the illness. We don't yet know the answer to that. But there is definitely strong support for the association of this gene with schizophrenia. And one report associating with autism. Autism is not by any means the same as schizophrenia, but there is some interesting overlap between the two disorders. Developmental delay, social impairment of social interaction, and several other genes reported to be associated with autism or schizophrenia have been shown to be associated with the other disorder. Now, what is this gene? It was originally described as the insertion site of a virus, of a helper virus, which is required for a virus called the Abelson virus to induce leukemia in mice. That is where the name comes from, the Abelson helper integration site 1. So the Abelson helper virus, which helps the Abelson virus, inserts itself at that point in the genome. Which of course is very exciting, ostensibly because the supposed connections between immune mechanisms of viruses and schizophrenia. So we spent a little bit of time thinking and looking along that route, but that does not appear to be the case. But at the same time, as we were studying this gene, as we came upon this gene in schizophrenia, another group of researchers was studying a very rare autosomal recessive condition called Joubert syndrome. Joubert syndrome is a severe recessively inherited developmental disorder. It's characterized by very clear abnormalities of the cerebellar peduncles, which causes them to look on MRI like molar teeth. It's called the molar tooth sign. There are other abnormalities of kidney. There's also cortical abnormalities. So this is a disorder, a recessive disorder caused by a mutation in the AHI1 gene. And this was discovered by the same route as we took, except these people were studying families with Joubert syndrome, doing linkage analysis, positional cloning. So what is clear is that if we are talking about an association of AHI1 with schizophrenia, we're talking about an association that is not an autosomal recessive and cannot be the same pathological causation as Joubert syndrome. One of the fascinating questions, which I don't have the answer to, is there more schizophrenia in the families of individuals with Joubert syndrome? And this is a very rare disorder, and it's never really been studied, and I don't know the answer. A lot of the families with Joubert syndrome have been identified in Saudi Arabia, and thus far no binational grants have been set up between Israel and Saudi Arabia, although one hopes that may happen in the near future. What about the gene? It's a large gene. It has 29 exons. It has 7 what are termed WD repeats, and this is a particular type of domain that is very important in proteins which are scaffolding proteins which serve as scaffolds, and clearly this can explain the involvement of AHO1 in brain development. AHO1 is involved in brain migration. I have a student now in common with Audie Reiner at the Weizmann Institute who's busy studying early development in fetal brain and the influence of AHO1 and AHO1 mutations on early migration of cells. So clearly this fits in nicely potentially with neurodevelopmental theories, that there may be a possibility that AHO1 may be influencing neural development, even individuals who do not have the kind of deleterious mutations that cause Joubert syndrome, and that these individuals might be subsequently susceptible to schizophrenia. I didn't say much about the C6-ORF gene. It's right next to AHI1. It's coded in the opposite, it's transcribed in the opposite direction. It's present only in humans, and we don't know very much about it other than that. Besides studying the genetics, we've been looking for convergent evidence for a role of AHO1 in schizophrenia. So this is an example of one of the studies we did. We were interested in whether the variants that we found influence expression. So we looked at lymphoblasts from our schizophrenia patients, and we see here that indeed schizophrenia patients do have increased expression of AHI1 as compared to control subjects. This is in lymphoblasts. These are the same patients from the Tiber clinic that I talked about earlier. And we also looked at brain expression, and the same variant that is over-transmitted to schizophrenia patients is associated with a higher level of expression. Carriers of the A allele of this particular SNP have greater AHO1 expression in brain than non-carriers of this allele. Yes?
You predict this in identical twins, which one is patient and the other one is not? You see differential expression?
Yes, if AHO1 is involved in schizophrenia in that particular twin, you certainly should see that. But because not all individuals with schizophrenia manifest these genetic abnormalities, we might not see it for that reason. But if AHO1 is involved in schizophrenia in those twins, then we should see this difference, yes. Absolutely. We've also started to work with AHO1 knockout mice. Now, obviously, this is looking in the other direction. We'd rather be working with overexpressing mice than with knockout mice, because the mutations that we have are over-transmitted to. But nevertheless, we thought it of interest, so we did set up a mice colony, and we're not looking at the homozygous mice, because the homozygous mice are severe phenotypes which resemble Joubert syndrome. But we are interested in the heterozygote mice. We've just started to work with them, and we got them from the mutant mouse center of NIH, and we started with cognitive tests, and it could be beginner's luck, but the heterozygotes do appear to have abnormalities of novel object recognition. These are the AHI1 heterozygous knockout mice, and these are the wild-type mice. So this is just the beginning, and we'll be doing a whole comprehensive series of studies with these knockout mice, and at the same time we're going to be looking at mice that overexpress AHI1 as well. So finally, I'd like to end off with some very much more recent work that is as yet unpublished, and that is the genome-wide association study we did. So talking about I talked earlier about the approaches, the methods one can apply in psychiatric genetics, And I mentioned that one can do that in the earlier years. One looked at particular regions because we did not have the technology for doing genome-wide studies. But now that technology exists, it's possible to genotype in a space of 2 weeks what used to take 2 years. And we did a genome-wide association study using a SNP chip from Illumina with 370,000 SNPs on the chip, and completed that recently, and it's now under consideration for publication. We used 53 families here. Genome-wide association studies are generally done as case control studies, where you have a large number of cases, a large number of controls. There are a few groups in the world that do family-based studies, where one has parents and children, and we prefer that method. We think it's much more resistant to population stratification effects. We analyzed the GWAS with standard methods. We corrected for possible spurious results due to statistical significance not being pegged high enough. We used what is called the false discovery rate. And these are the results of the GWAS, so the Genome-wide Association Study. This shows the numbers of the chromosomes along this axis, and this is the cutoff for genome-wide association. And as you can see, there's several genes associated with schizophrenia other than the chromosome 6 region, which is here. The strongest is actually on chromosome 2q, which, if you will recall, is a region that we showed linkage in our first study done published almost 12 years ago. This is one of the genes in which we found association in the genome-wide association study, and now these are all on chromosome 6, on chromosome 2, and one significant finding on chromosome 6 in the region of the AHR1 gene. So this shows the data. It's a bit busy, but this shows the SNPs that were significant, the genes in which they were, and this is the one from chromosome 6, and all the others are on chromosome 2. In genome-wide association studies, the demands have increased considerably. One has to publish replications along with the original report. So I'll come back to this. We immediately sought replications in 2 different samples, Jewish-Israeli sample and a sample of German schizophrenia patients, several thousand patients and several thousand controls from the Munich area of Germany. And we had one SNP that was significant in the Jewish-Israeli sample as well as in the Arab sample, and 2 that were significant in the German sample as well. So these were not in the LRRF1P gene. One was in a gene called ACSL-KCNE4, and the other one from the Munich sample were in other genes. But we do have replication, so that is, I think, a critically important finding. What about the strongest gene? The strongest gene to emerge from our analyses was a gene called LRRF1P1. The joke in the lab is that it's actually the Lerer gene, LRR, and it's a transcriptional repressor that may regulate TNF and EGFR, and both these are proteins which have previously been associated with schizophrenia. And this is a simplified representation of the interactions among the various genes and the possible role of LRRF1P1. So that is the stage we are at now. Besides AHR1, which remains our favorite and which we will be studying more and more at the level of neurobiology rather than the level of genetics, our aim there is to establish the role of AHR1, a possible neurobiological role of AHR1 in schizophrenia, based on our assumption that this gene is associated with the disorder, at least in a subset of patients, and if we can understand the pathophysiology, and how it's potentially involved, we can understand the pathophysiology of the illness, at least in some of the patients. And we'll be doing the same when we have the resources with these new genes that have come up from the GWAS, particularly those for which we have replication, and once the paper is published, for which other groups replicate as well. This is another one of the genes. I won't go into that. So what we're going to do in the future, as I said, replication studies, functional characterization of the implicated genes. We'll work in cell models and in model organisms. And we're also going to be sequencing. Sequencing is the key. We're sequencing the genes, we're sequencing the regions. We're looking for possible causal variants. Variants that may influence the structure, either exonic, which is unlikely, but more likely variants which are located in regions that influence expression in the 3 or 5 prime regions, or in regions adjoining the gene which influence the expression of the gene. And we're also doing de novo sequencing, in other words, starting again and sequencing not large samples, but sequencing families, looking at individual families, and based on the hypothesis that there may be a very large number of genes that are involved, and we might find a gene involved in 1 or 2 families, and that might be enough for us. So that's how we're going to be keeping ourselves occupied for the next few years. And this is my group. The person principally involved in this, in the work I presented, is Anna Alkali, who's a PhD student, now a postdoc elsewhere. This is Kyra Kanyas, now Kyra Sana, who helped actually collect all these samples in Thailand, and had a car stolen twice in the process, but she kept going back and put these families together. Yoav Cohen, a psychiatrist, who works a lot with me on this, and several other members of the group who do other productive things, but not necessarily related to this project. So thank you. If there are more questions.
Other questions? So it looks like there are a lot of diseases, and the question is it possible to make better diagnostics? So like to differentiate the different types of schizophrenia and then check, like psychiatrically, and then check.
That's a very important question, a key question. Most people agree that schizophrenia is not a single entity. It's a single entity today because it was agreed in writing the DSM that at least if we're going to be wrong, we should all be wrong in the same way. And but clearly it's a heterogeneous disorder. But it's not been possible. Attempts to create or to differentiate diagnostic subgroups have not been successful. They've been done in many different ways, statistically, clinically. It's not been possible to differentiate subgroups. So, to look for genetic correlations with these subgroups. The subgroups that have been created have not had validity, even at clinical level. And so, the hope is that the neuropathology or the genetics or the can lead us to the subgroups and not the other way around. But it's clearly a huge problem because we're looking for genes associated with an illness which is not one illness. So I think your question is really very much on target.
So a related question, is there a continuum from being normal to being...
Clearly there is a continuum. There is a type of personality disorder known as schizotypal personality, which people tend to be strange. They tend to be to have sort of odd ideas. They tend to have difficulties with interaction. They work a lot with computers. There is definitely
How do these people are related to autism?
There is definitely a continuum. There is also maybe a continuum between schizophrenia and bipolar disorder. I personally don't believe there is. I think it is a question of assortative mating in that. I think that, but if one takes a person, a classical person with schizophrenia, and compares that person to someone with classical bipolar disorder, If you compare, what's the name of the actress who's married to Michael Douglas, Catherine Zeta-Jones. If you compare Catherine Zeta-Jones to one of the ladies hospitalized chronically in Krakow, there's no comparison between them. But between those two extremes, on the bipolar on the one hand, there seems to be a continuum. And there are people who have what is termed schizoaffective disorder, which is where they're schizophrenic, but they also have mood swings. And a lot of that could be due to what is termed assortative mating, and that is people with psychiatric disorders tend to marry people with psychiatric disorders. And in some communities, for example, the Haredi community in Israel, that kind of marriage is actually directed. Shadchanim will ensure that someone who has a psychiatric disorder marries another person with psychiatric disorder, not someone without a psychiatric disorder. And there's a growing awareness of the fact that this is a huge mistake. So you do have a continuum. The continuum could be an artifact, but it is present. And the continuum of schizophrenia and so-called normality certainly exists. Yes?
Some basic question. In the beginning of your talk, you said that now there is no question that it is brain disorder or something, although there is no pathology, which is clear.
Yes.
What would be the alternative? I mean, is there any psychiatric disorder which is not brain disorder?
That's an interesting question. The assumption is that it is a brain disorder. Yeah. There is no brain pathology evidence. So what is the alternative? The alternative is that it is I'm not saying that any kind of behavior has a brain substrate, obviously. So I'm not I'm not just relating to it on that level. But the psychoanalysts, for example, had the theory that schizophrenia is a type of aberrant behavior which is induced by parenting, that the parenting of the children induces a kind of behavior in the children which is schizophrenic in nature. And that was the psychoanalytic theory which prevailed for a long time. There are psychiatric disorders which have causations which are not entirely from within and not entirely endogenous. Take, for example, post-traumatic stress disorder. Individuals with post-traumatic stress disorder, let's say soldiers who've experienced severe trauma in battle, they do not have pathology prior to the emergence of the PTSD. I've seen people like that. I did. Pardon?
So the external event that triggers.
Yeah. So here you have an external event that triggers a severe psychiatric disorder, and not one which you can ascribe primarily to brain pathology. You could say there's an interaction between latent brain pathology and the external event, but there's no question that the external event is a key factor in causing the disorder. There are other examples like that but PTSD is probably the strongest one of all. So it really depends at what level you're talking. But if you were to put forward, and I was afraid you might be saying that if you say it's a brain disorder, where is the disorder? The answer is that you don't have the disorder. You don't have the ability to pinpoint the precise pathology.
But it's not clear,
yeah.
Or maybe diffuse.
Right.
Back to our family. Can you say anything about the specific schizophrenia subtypes that express in them relative, let's say, to this unique sample that you described? In the end, are these different? Since you have such a uniform, genetically uniform...
yeah, one thing I can say, and that is striking, and that is that the incidence of affective illness means mood disorders, depression and manic depression, in the families from the Taibe region is very, very low. We have a lot of Jewish families. You find a much higher incidence of individuals who have mood disorders in the Jewish families besides the schizophrenia patient. In other words, you recruit the families because you're looking for families with schizophrenia. So your cutoff is you want at least 2 children with schizophrenia. You then go and examine the brothers and sisters, you examine the aunts and uncles, the grandparents if they're around, and you build the family. So you'll find there's no question that there are many more patients with mood disorders in the Jewish families than among the Arab families. And the question is whether this is a reflection of the genes which are at work, or whether it is an issue of assortative mating. I don't know. There is much less assortative mating among the Arab families. I personally interviewed and examined several of the families, and what is striking is that there's a much higher level of social acceptance. The individuals with schizophrenia are socially accepted. Many of them marry, a lot more marry, and a lot more have children than in the Jewish community. The cutoff is violence or disturbing or socially embarrassing behavior. In other words, if the person behaves badly in public, attacks people, takes off his clothes, curses, then he will be considered ill and taken to the clinic. But if he's just a little bit crazy and cut off from the rest of the world and occasionally sits in a corner and talks to himself, that is socially acceptable. I'm not decrying it. I think it's a reflection of an approach which the rest of the Israeli society would do well to consider. When you consider, that, for example, attempts to establish a hostel for chronic schizophrenia patients in several areas of Israel, in several cities, encounter enormous opposition. People are all in favor of having such hostels, but not in their, what do the Americans say, NIMBY, not in my backyard.
Do they work? Do they work?
People with schizophrenia, in a much lower proportion than the rest of the community, and in a much lower proportion than people with other psychiatric disorders, like bipolar disorder, for example. Generally, chronic schizophrenia patients are employed in protected kind of work, or in what is it in English? Rehabilitation type work, and not on the open market. It's unusual to find patients with schizophrenia who are able to be employed on the open market.
There are no more questions. I think I'll stick again.
Thank you.